Okay, this is, as Lauren says, one of the tools that's been developed within an Interreg program called DIESE, which is one of the contributing projects to 3C. And it builds upon our work using this estuarine clam, Scrobicularia plana. It's a very common clam in most European estuaries. It's important ecologically. It's one of the major food sources to a lot of birds and fish within estuaries. We know quite a lot about it biologically. It burrows in the surface sediments, creates these star-shaped feeding marks, where it's got little siphons that come out and feed over the surface of the mud. And it's a very good biological indicator of sediment quality on different levels, from metals, organometals, and other pollutants. So this tool, looking at endocrine disruption, is another feature for which we can use Scrobicularia. I'm not going to spend too much time on this, but endocrine disrupting chemicals are a lot of exogenous agents. That means they come from outside of the organism's body, and they interfere with production, release, transport, metabolism of natural hormones in the body responsible for their normal development. And of course, if that process gets perturbed, then we see problems. This species is what's called gonochoristic. So normally you get males and females. So this is a microscope preparation of a male gonad, the testes, with sperm. These are oocytes in the female. But we also find this condition of intersex, where you get egg cells occurring in the male gonad. And as we now know, that is not the natural situation. This is an abnormal situation. And furthermore, we can scale the level of impact. So we can scale the level of feminization of males, and it is a feminization process. It is alteration of the males into females, and we can have an index of severity that we can measure. And all this is done by simple microscope preparation. You don't need a lot of fancy equipment to do this. You just need a bucket and a fork, go out, dig some clams, dissect them, and look at them under 100 times on a light microscope. So it's a very simple process to do. Within the DIESE program, we sample clams over 3 years from all around the Channel coast. Did 108 sites in total. So we've got a good spread around the Channel coast, and it really was that sort of broad brush approach. Is this a widespread problem? Does it occur in specific areas or not? There's a subtle detail on this map, which again is a Google Earth map. The green sites is where we find no effect. The red sites are where we find intersexed individuals. And I'll say a little bit more about that in a minute. So looking at, if you can imagine that map, this coastline stretched out into a straight line, and the same on the French side, we end up with a map of that, or a graph of that sort of format. So this is the English Channel coast, this is the equivalent French coast, and they're more or less opposite. What we find is that in the UK, we found about half of the sites were affected, so we found intersex clams at just over half the sites. 70% of sites affected on the French coast. So these are the percentages of males that are feminized. So up to almost 50% on the Severn, over 50% on some of the French sites. We could also look at that in different ways, look at cumulative severity on that scale of impact. We could look at sex ratios and find significantly skewed, either masculinized or feminized populations above what we would expect normally. We can also look at accumulated contaminants in the sediments at those sites as well, and whether they are compliant with sediment quality guidelines. This is one for cadmium, where in most instances levels are below the guideline levels. And we can do that with the clams as well, looking at contaminant levels in the clams. We have an existing mapping program to visualize that data. This is an example of copper in seaweeds. It's very old now. It's an old bespoke program that was written in the 1990s, gives you a static image. and there's all sorts of problems with it, with things overlapping. So we've now got a great interest in using GIS as a better presentation tool for a lot of this multivariate data that we have. Just to put this into context, I think for the catchment-based people, where do these chemicals come from, the endocrine disruptors? Well, they come from sewage, but also from agricultural sources. Basically, any vertebrate produces a lot of these hormones. Synthetic chemicals can mimic their activity, and other endocrine disrupting chemicals are widespread, including detergents, plasticizers, and a lot of ubiquitous contaminants also have endocrine disruptive effects. It was first spotted in fish, freshwater fish close to sewage treatment works, but we now have a lot more evidence to suggest that agriculture can be a source, and this particularly came from the Avon estuary in South Devon, where there is an oyster farm. And this was looking at the effect in clams over several years between 2004 to 2011, where we saw a gradual decline in the incidence of intersex in male clams, and this coincided with changes in how cattle were being managed within the catchment. And this was driven, as Nick was saying earlier, a sort of a side effect of looking at one problem, because this was driven very much by the oyster farmers in the Avon estuary. They were failing their coliform counts in oysters, hence they were putting pressure on the dairy farmers to manage their cattle in a different way. By moving them away from watercourses, fencing them off, we had reduced incidence of defecation, urine getting into the watercourse, which was good in terms of coliforms, but it also seems that it had a beneficial effect in terms of the intersex in clams. Although that is purely correlation, we have no cause and effect data, no firmly linked data for that. But it just serves as an example to show that, you know, possibly there's a link between how livestock is being managed within the catchment and what we see down in the estuarine end. So I think that's my 5 minutes.
Brilliant.